Do you want to share the first
Yes.
screen in the meantime?
Yes.
Go ahead if you want.
It's okay? Can you see it?
Yes, perfect.
Okay. Well, Leonora, when you decide, we can...
No, it's up to you, Andrea. Whenever you want, just start with the presentation. Thank you.
Well, okay. So we will talk a little in Spanish now, Soren, for 2 minutes only. Bueno, vamos a empezar entonces a tiempo porque es tarde en Dinamarca, entonces para no terminar tan tarde, les damos la bienvenida a todos. Realmente es un gusto ver la cantidad de gente, como recién nombró Eleonora, muchos que de nuevo se han unido. Muchísimas gracias por acompañarnos en esta nueva charla, que es la quinta dentro del ciclo de conferencias virtuales que estamos organizando desde nuestra sociedad, SAMIGE. Y antes de comenzar y presentarles a nuestro invitado de hoy, bueno, les iba a solicitar como siempre que mantengan silenciados sus micrófonos y que apaguen sus cámaras. Y respecto a la dinámica, como siempre, como en ocasiones anteriores, las preguntas las vamos a escribir en el chat, que va a quedar habilitado durante toda la charla, y al final vamos a armar la discusión, en forma ordenada vamos a ir planteando las preguntas de acuerdo al orden en que se han hecho en el chat. Y al finalizar esa discusión, también como siempre, les vamos a pedir que se queden un minuto, si puede ser, para que vamos a prender las cámaras y sacar algunas fotos para que nos quede de recuerdo los quienes nos están acompañando hoy. Y bueno, y nuevamente agradecer a Matías Canepa, nuestro host del CCT de Mar del Plata, que siempre nos asiste con tan buena predisposición. Muchísimas gracias, Matías. Y de nuevo también al doctor Leonardo Curati, que por su gestión que nos ha permitido y ha facilitado el uso de la plataforma Zoom. Well, I'll switch to English to introduce our guests to you. It's a great pleasure for me to introduce Dr. Soren Molin, one of the world's most remarked references in microbial adaptation and evolution, particularly focused on the opportunistic pathogen Pseudomonas aeruginosa in cystic fibrosis infections. His authority in the area is based on a prolific and remarkable scientific and academic career, which has covered broad aspects of microbiology. Among other contributions, his lab was one of the pioneers in the bacterial biofilm research area, introducing innovative molecular tools and image analysis that were key for a better comprehension of bacterial biofilm development. These activities are still the foundation for his ongoing science, which also comprises cell-to-cell interactions and evolutionary processes, and have greatly contributed to the development of the field through detailed studies on microbial adaptation and evolution in cases of chronic infection. During the last decade, Soren has been attracted and actively engaged in translational studies of long-term bacterial infections in human airways, a topic that he'll certainly address in his talk today. All his contributions have been reflected in around 300 publications, many of them in the most prestigious journals, reaching, according to Scopus, an H-index of 93. Following all these huge, following all this huge work, he was rightly been awarded by several institutions and foundations. Soren has also been engaged in teaching at all levels at Danish University for 40 years, and currently he is professor and group leader of Novo Nordisk's Foundation Center for Biosustainability in the Technical University of Denmark, wherein he was the director of the scientific section from 2011 to 2015. Last, but certainly not least, I like to mention that all his quality as a scientist is as great as his quality as a human. Many of you know Soren. He had visited us in the SAMIGE 2009 in Carlos Paz and in the SAIG 2012 in Mendoza, where he could enjoy our Malbecs, of which he is a passionate expert. He's a great host. Everybody who had the possibility to visit Copenhagen enjoyed and loved the place, and mostly because of him. Soren is a very generous person, also in science, since he's always open for collaboration and for sharing knowledge and facilities. He's constantly innovating and brainstorming ideas. Consequently, he transmits his passion for science with great enthusiasm and devotion. Working with him is without any doubt a fulfilling experience. Soren has mentored more than 50 PhD students and more than 55 postdocs, but I'm sure his legacy extends that. He has encouraged many of the following scientist generations to the passion for microbiology. My students that experienced being in his lab are witnesses. Today, Soren is going to talk about life of Pseudomonas aeruginosa in the airways of cystic fibrosis patients. So, as a friend, I'm very happy, Soren, that you are here. It's a real honor for us, and I'm very grateful to you for accepting our invitation. So I'll stop sharing my screen now in order you can share yours, and the audience is all yours. Thank you very much.
Thank you very much, Andrea, for your introduction, and whatever you said is really appreciated and returned. And I've enjoyed your hospitality several times coming to Argentina. And I just think it's a pity that I'll be there physically today. But don't worry, I'll take revenge and I'll be there someday. So thanks a lot. Thanks for the invitation. It's always a pleasure to meet not only Andrea, but also people who have been seeing my lab and whom I've met when I came to your country. Now I'll see if I can do this trick of getting, yes. Can you see my slide? That's good. All right, so as Andrea said, we have for some years been doing different things. The work with biofilms, which started in the late 90s, were mainly initiated to start interactions between organisms. So when everybody were growing single species, single strain biofilms, we started out with 8 different strains from a water rinsing device in Denmark, and realized immediately that it was absolutely impossible to get any sensible things out of that. But during a sabbatical in California, I met, before that, I met Professor Niels Højby at the main hospital in Copenhagen, and he was telling about a collection of bacteria which they had pulled out of patients suffering from cystic fibrosis. And I just want to give you a brief, what do I do here? Hang on, now I have to just see how I get the, how do I shift the slide? Here we go. So I need a little bit of background. It's very short. Cystic fibrosis is a genetic disorder. It is a change of a gene in chromosome 7 called the CFTR gene. And the consequence of mutations in this gene is that the mucus in lung go from a fluid mucus, a fluid mucus where the bacteria can be swimming, and because of the ciliae that go in one direction synchronously. Whatever bacteria and particles and fungi come down through the respiratory tract, be brushed up through the respiratory tract again, they don't have a chance to colonize. But for patients, this mucus is very, very viscous because of water dehydration. And this is part of the consequent mutation that is affecting a chloride channel, a chloride pump in the lung epithelial cells. So lo and behold, what happens here is that in the lungs of the CF patients, because of this mechanic defect of the ciliate, as you can see here, the ciliate are really compressed and they are not able to do this clearing. Also, the mucus layer is very viscous. So eventually, many different bacteria and also other strange organisms will actually colonize the mucus of the lungs of the CF patient. There are other consequences. For example, you do find in pancreas and also the bile, you find mucus that blocks transport. But what people with CF die from is mainly lung failure at the end. And this, of course, is a very horrible disease. And in contrast to what people normally, it's not because the bacteria are infectious, it's not toxins or pathogen or virulence factors from the bacteria, but the problem is that the immune system of the patient reacts very aggressively to the presence of the bacteria, and therefore, in fact, the lung tissue is destroyed by the immune system. So this is enough introduction, because what is interesting for us is that there are bacteria and there are certain bacteria which we are interested in the lungs of the CF patient. And because the bacteria are sitting in an environment which can easily get coughed up by the patients, we have access to samples from the patient, so we can study which bacteria and which other organisms are in the lungs. So this is just a 16S ribosomal RNA profiling of the population of bacteria in a couple of young patients. And I don't want you to read any of this. It just shows that there's a huge diversity of bacteria in the lungs of the patient. So as I said before, they basically can all, if they have a chance to grow at 37 degrees in the substrate that the lungs produce, and then they have a chance to colonize. And even in young children, you have this enormous diversity of bacteria. But in some cases, as you can see here and here and here and here, there seems to be one species which is dominating completely. But then later on, it disappears again. Here it's reduced. You only see very, very little, and then it comes back. And of course, the reason why we have this changing composition, as you see here in this kinetic graph, is that the patients, once they are diagnosed with CF and with bacteria in the lungs, they're being treated with antibiotics. And that's why we see these cycles of appearance and disappearance. Here you can see from the bottom that which specific genes are present in the lungs. And the one I pointed out before, this dark purple, is actually Pseudomonas aeruginosa. And what we decided to do a long time ago, and all many medical doctors decided to, was to focus on Pseudomonas aeruginosa, because it is one of the dominant organisms in the lungs. So this is our model, and this is our research focus point. So basically, we investigated particular aspect of Pseudomonas aeruginosa, which is an environmental bacterium in many different places in the environment, in water, associated with plants and soil, whatsoever. And from time to time, these bacteria can enter human beings, not just CF patients, but also other types of disabled patients. And in the case of CF, they simply move from the environment into the lungs of the patients. I also want to point out that in the sinuses of the skull, of the head, we also find the same types of Pseudomonas aeruginosa strains that we have in the lungs. I'm not going to deal with that anymore, but it has turned to be an interesting traffic there is between the with our nasal sinuses and the lungs. So in short, we have decided many years ago to study the migration of Pseudomonas into the airways of CF patients to simply find out the biology of this process. So we know what Pseudomonas aeruginosa is. It lives in the environment. The question was, and still is, what does it become after transfer to the airways? And the most important message I have, and that has really been very hard to convey to medical doctors. So the message is that the CF airway is an ecological niche, which is not a test tube. So therefore, all the studies that you do in the lab, both in biology labs and in the clinical labs, where we take the bacteria from the patients, grow them in LB, in test tubes or flasks, will always be very, very far from the real environment. And the real environment is a complex ecological niche where you have nutrients produced by the lung cells. You have the immune response, which immediately reacts to the presence of the bacteria. You have, of course, medical doctors that keep bombarding the environment with antibiotics. And all things together give a huge number of different selection pressures. So when we talk about selection, it's not just antibiotics. I think that was a mistake made by many people in the very beginning of this era of studying CF bacterial populations, that everybody thought that antibiotics was the only selection pressure which was worth dealing with. And I think you can see here from this complex picture that obviously there are many other selection pressures which will also form the shape of the bacteria, will also bring about changes or select for changes that happen in the bacterial population. And that has turned out to be the real obstacle. So in order to really understand what's going on, we take into account that there's not just one or two or three selection pressures operating on the population, but there's many, many more. So one thing which of course is a medical problem, which is also a very interesting biological problem, is why do persistent infections persist? In other words, if you have a child with CF who is infected with one particular clone of Pseudomonas aeruginosa, and you then follow this patient for the next 10, 15, 20, 40, 50 years, in many cases, it turns out that exactly the same clone is still present in the particular patient. And what is accomplishing this amazing persistence of some of these bacterial clones? It's not in all patients, but in many. So we decided some 5-6 years ago to do a dual investigation of this problem. First of all, we wanted to look at the genomes of the bacteria, particularly in patients who had the same clone for many years. So we did genome sequencing. We started in 2013-14, and we thought very naively that all we had to do was to read the genomes, and then we would figure out exactly which patients caused the persistence. And then the medical doctors could get molecular markers, genetic markers, and then they would be equipped with a wonderful diagnostic tool. And of course, it didn't go this way. So when we really had banged our head against the wall many, many times, not being able to understand the genomics, we decided to do a phenomic analysis. So we went back old-fashioned phenotype analysis of many, many, many isolates, exactly the same library of strains that we had from genome analysis, and that was about 500 isolates from 30 different patients. And then we took all these isolates from which we had the genome sequences and then took them through a lot of phenotypic analysis. Basically anything which was clinically relevant and also which could be performed easily in microtiter plates to make it easier for the poor guys who had to do all this. And then finally, we had to put all this together, and we're still struggling integrating genome data and phenomic data, but we are coming better. We're actually doing something which makes a lot of sense. So on one hand, compare the phylogeny of these different ways based on the genome sequences, and on the other hand, we can do some modeling of the phenotypes, and we have chosen to work with a special type of model which is called archetype analysis, which has been very, very useful for our analysis of many different phenotypic traits, and I'm coming back to that later. So this double perspective investigation has been what we have been doing over again for 5 years, and I'll give you just summary slides of what came out of it and why it has led to a new program, which I will present at the very end. So from genome sequencing, we can get, of course, many different types of information. So first of all, when we take isolates from the different patients, we can, in each patient, we can see how many different clones of Pseudomonas can we find. In many cases, it's only one, but also in quite a few cases, we find more than one clone. I should say that all this information has been mainly based on the strains that are out in the clinic. And in the clinic, technicians are taking isolates of Pseudomonas from the samples, from the sputum samples, and then they store X number of different isolates. But they are not seeing the real diversity. So no matter what I say about monoclonality of these, take it with a grain of salt, because we know now that even though we have a dominant clone, which may constitute about 95%, we will still have subclones of different types of Pseudomonas, which are very different from the dominant one, and which only constitute maybe 0.1% or 0.5% or 1%. So we don't see them in the library that was made in the hospital. But we are considering, and we have been doing a little bit of analysis of the entire diversity of the population. And at the end of the day, this is becoming very, very important. What we also see from the genome sequences over here is that each of the different clone types will have variation because of mutations that are accumulating in these persistent populations of bacteria. So if you look at isolates longitudinally, which we have been doing from different patients, we can see that more and more mutations are accumulated in the genomes. And then, of course, we can try to make sense of these mutations. In most cases, they don't make sense, but I'll give an example of that later. Then, of course, you can read about virulence factors. It's still an open question if pathogenicity factors of the bacteria play an important role, especially in the beginning of the infection of the patients. And I would not be able to say for sure that it's not important, but we know that very soon after infection, many virulence factors disappear. So for sure, they are not necessary in the long run. They're not required for persistence, but they may actually play a role in the very first period of infection in the CF patients. Then we can look for genes that have to do with antibiotic resistance, and we can look for mutations, and we can see if persistence is associated with antibiotic resistance or not. You have, in Andreas' group, a long-standing experience with antibiotic resistance, and you can really read a lot of this when you look into the gene sequences. It's not the only thing that gives persistence, but of course it is a very important thing. when the patients are treated with antibiotic all the times. And then finally, you can look at epidemiology. You can see from where are these clones coming? Are they all coming from the environment, or are they coming from other patients? Are they transported from nation to nation because people travel? And this is all very interesting and also indicative of maybe some clones being more potent in creating an infection in the patients. So I just want to give you one case of what we pulled out, the 500 strain genome sequences. And what here is mutations in genes which seem to be hit again and again and again and again by mutation. In other words, there seems to be a selection for these mutations because we find much more frequently than you would expect statistically, we find mutations in these genes. And in the young kids that from where we have all these isolates, it turned out that there were about 50 different genes in Pseudomonas that seem to acquire mutations or at least accumulate mutations much more frequently than others. And when we put it all up in this frame, you have all the different clone types here, stress of Pseudomonas, and then you have the 50 genes which are very frequently mutated as we see it in the isolates from the patients. And when you go down and see, is there some specific constellation of mutations which seems to be typical for persistent infection, the answer is clearly no. There is no pattern which makes sense whatsoever. The reason why we have many more mutations over in this part relative to this part is all these patients here, they're much younger. So they haven't had Pseudomonas for more than a very short time. What we could then do was to see, are there special pseudocap functions which are hit? And of course, we find resistance mutations. That was not surprising. We knew already that regulatory mutations, that is genes which encode transcriptional regulators, they are frequently hit by mutation. And then we found mutations sometimes involved in biofilm formation. So some surprises and some not, but the pattern is not existing. So we couldn't make sense of this. And we couldn't say that if a strain gets mutation, it has a higher chance of persist. It's simply not possible. That. I think what is much more likely to come out eventually is that combinations, specific combinations of different mutations in some of these pathoadaptive genes might enrich for persistence more than others. But it's far too early to say that. We need many, many more sequences, and then we need a machine to handle all the information because we are not able to do it ourselves. So from this, we could say that many important properties securing pathogen persistence are multifactorial. That's what we still think. We have a lot of interest between mutations, and many factors are needed to secure the persistence of the bacteria. And we also know that it's not just one trajectory which results in persistence. There must be several different adaptive trajectories that pave the way for the persistence of the infection. And therefore, genomes must be complemented with relevant phenotypic data. And I'll just give you a few examples of that. Can you hear me still? I hope so. I have a message saying my internet connection is unstable. I hope you can see this. So I just want to give you an idea of what this archetype analysis is. So basically, we take from our phenotypic data, we look at different combinations of phenotype and ask if they appear frequently in the population of the 500 isolates. And then you see what we then do is how many different combinations will still separate the whole 500 strain collection into different defined groups. And in our case, with the phenotypes that we had measured, we could identify 6 different archetypes, and they come all the way here. And if we added more combination, really nothing much happened in terms of grouping. So we set a barrier by the 6, and then we divided all the isolates in one of these 6 archetypes. And the pattern you have here is that all the strains which cluster over here, they are fast growing, they are antibiotic sensitive, and so forth. So they represent the early stage of infection. They exactly in their phenotype like strain on the outside. If we go over in this part of the projected archetype analysis diagram, is a multidisciplinary diagram, of course. We find features which are usually associated with chronic infection, slow growth, ciprofloxacin resistance in this case, and also increased adherence to surfaces indicating strong biofilm information. So with this, we can easily divide our population or subpopulations or cohorts of patients and look at this diagram, this archetype analysis, and then we can ask specific questions. And we did that in one case here where we asked, when do we see a migration towards the adapted phenotype, which is over here? And the answer was very simple. If we compared first isolates with 2 to 3 year old colonization, already after 3 years, we need to see a migration towards the adapted state. So we concluded sort of generally that it doesn't take 10, 15 years for the bacteria to adapt. They are very much on their way to become fully adapted after 2 to 3 years of oxygen in the lungs. That tells the medical doctor that they shouldn't sit down and smoke cigars, because they have, as soon as they realize that Pseudomonas aeruginosa is there, they have to identify traits which are usually associated adaptation. And I just mentioned a few before. Ciprofloxacin is one, but that's mainly because not treated fluoxacin very frequently. The other one, which is so dead simple, is growth rate. And it turns out that growth rate is rapidly reduced among the populations in the different patients. So we are advocating now to the hospital that on top of all the antibiotic susceptibility measurements they're doing for their diagnosis, why don't they measure growth rate of these when they have them isolated in the lab? And if they see some kind of dominance of slow-growing Pseudomonas aeruginosa, it's a very good indication that this patient now at the point of being chronically infected. And we'll test a large number of patients to see if this is true. But if it is true, if there is a strict correlation between reduced growth rate and fully adaptation, then of course it's just a simple extra parameter to test, and it may be much, much more useful than antibiotic susceptibility test, because as you'll see in a minute, it really doesn't very much information because it's all done in the laboratory. So just to summarize this first part, I just want to argue that if we have clinical data, which they determine in the clinic, like antibiotic resistance, like growth rate, like any other suggestion we come up with, and we have the genomic data, we can begin not only to take the clinical data, the sequence data, and try to put it into a database and then make sense of it, but we can also begin to draw and mine from public databases, those epidemiology databases and biomedical databases. We can, after cleaning the data, we can begin to integrate all this. And for that, we're pretty smart data analysts. And we have a couple in the neighborhood in Copenhagen. So we're beginning now to let these brilliant bioinformaticians and modelers trying to see how far can they come with data integration. Because if they can integrate the clinical data, the sequence data, and data from the outside hospitals in the world, and then get some consistent pattern coming out of that, they can translate that information back to the clinicians. The clinicians should be able to exploit that information in changing their diagnosis or changing their therapy. That's the whole point of this anyway. And this, of course, wishful thinking, but we will hopefully get a huge amount of money to set this system up so we can actually do that with an enormous labor force in the future. So then the question is, when it comes to phenotypes, of course, we can check 5, 6, 7, 8, 10 different phenotypes in the lab. It takes forever. So we decided at some point, instead of doing that, we wanted to read the global gene expression in Pseudomonas aeruginosa when it's growing in the airways. And the trick here is that you send your postdoc who wanted to do this crazy stuff. You send him to the hospital, and then he has to be there when the patient comes into the clinic and delivers a sputum sample. And then he has to have an agreement with the medical doctors and the nurses that he can get a sample coming directly out of the lungs of the patient so he can fix it and make sure that whatever is there in terms of RNA is maintained and stabilized. Because as you all know, messenger RNA is really, really not easy to handle in bacteria. It decays with a half-life of often less than 1 minute. So with a fresh sputum sample, then you can begin to process this, and you can analyze it directly. So you can take this source in vivo sample, and then you can do RNA sequencing directly. Or you can isolate, you can isolate bacteria from the sputum sample, you can grow them up, and then from the same sample, you then grow them in vitro, and then you can do RNA sequencing. And then, of course, eventually you can compare the RNA transcriptions from in vitro experiments to those that you identify indirectly in the sputum sample. And this is what Elio Rossi did. It took him quite a long time to actually set up the method because it's extremely difficult. So Pseudomonas aeruginosa RNA is such a minor part of all the RNA you have in the sputum. So you have to be really, really careful with the way you analyze. I'm not going into details here. Since it's published, you can read how difficult it actually is. But the interesting thing that came out of Elio's work was mainly that if you look at four different patients, one with DKO1, one with DKO1 plus 2, one with DKO2, one with DKO2 plus an unknown, and take sputum samples from each of these patients with different bacteria, different infection times, different extra bacteria and microbes in the lung. They have different communities, different microbiota. And then you look at the overall transcription pattern in a PC plot, in a principal component analysis. To our big surprise, it turned out that all the sputum samples from, this is in vivo, all the sputum samples from the different 4 patients with different strains, with different mutations, the genetics of those strains is quite different, but they still clustered. So it looks as if it's not dependent, the way transcription profile looks is not dependent on the genomic content, but it's much more dependent on where the bacteria live. In contrast, the in vitro analysis from the same samples, growing the bacteria is pure cultures in vitro, then we see a completely different position of the cluster, and we also see much bigger. So this, I'm going to skip. So this told us that the core, and we focused on the core genome. Core genome in vivo expression is determined by the lung environment and its composition. So the environment sets the transcription pattern from the bacteria, despite the many different genetic differences, despite all the mutations that we know from the sequence, from the genome sequence, are present in the different isolated patients. So first of all, community structure plays only a minor role. In fact, we don't know that what the community structure is, but we know they're different because they check that in the clinic. And we can see that some patient has a lot of staphylococci, other patients have Achromobacter, yet again, other patients only has Pseudomonas aeruginosa, and seem to impact on the in vivo expression of the core genome. The other thing which comes out of this, of course, is that gene expression markers then may be useful in adaptive states of the infecting bacteria. So we hope that this pattern of gene expression, and I don't have time to go into this, but we certainly hope that this kind of pattern from these well-established chronic infections, and even before that, if we take younger patients, that there will be certain classes of genes which are induced or repressed, simply, and we expect that to happen simply because environment that seems to determine gene expression, not the genome sequences. So that's, of course, very important. So metabolism. We very early suspected that metabolism was a problem. That not a problem, but that it would be changed. And Ruggiero La Rosa, who is another postdoc in the group, they're all Italians. I don't know why, but that's what it is. Adaptive change of metabolism. We took from the same patient 3 different clones which were stably colonizing the patient. DKO1, which is an old clone, you just saw it mentioned in Elio's work just before. Then we have an intermediate clone, which has been there for some 5, 6, 7 years, intermediate. And then we have a clone in the same patient called DK15, which is completely naive. It just came into the patient a very short time before we took the samples. And then we grew them. And what is remarkable here is that DK15, which is the naive strain, grows very fast. The intermediate one, DK53, grows with intermediate growth rate, and DK1, which is the old chronic strain that many patients have for 40 years, grows very slowly. So as I said before, growth rate is a wonderful parameter, and it really shows, as by example here, that early infection stages are associated with fast growth, late infection stages with slow growth. What Ruggiero did was simply to screen for exometabolites. So he grew the cells, and along the growth curve, this is PA01, he grew the cells, took samples, and then he removed all the cells and just looked which secreted products he could find in the medium, in the substrate. And he did that all the way along, 15 to 17 samples per growth curve. And he did that for PA01, he did it for the early naive strain, he did it for the intermediate strain, and he also did it from the old chronic strain, DK01. And it doesn't take a long time to see that if this is the fast-growing wild-type pattern of excretion of different compounds, then when you go to the naive strain, it's almost the same. It's only a few that fall off the line. But if you go down here with the intermediate and especially the chronic strain, they're totally different. It's as if they are not Pseudomonas aeruginosa anymore. What he then did was to take those strains and then try to see if he could revert slow growth to fast growth. And here, is what comes out. This is PA01 growing in a robot where there is a selection for fast growth. There's no drugs, nothing, just LB. And as you would expect, there's some scatter, but there's no trend of PA01 growing faster than what you know from any lab strain of PA01. But if you take the early infecting naive strain, it very rapidly, he took deliberately one that was not growing so well, but it immediately came up after just a couple of hundred generations. It reached the same level of growth rate as the PA01 reference strain. With the intermediate one, it was much, much more difficult. And if you extrapolate this, it would have taken thousands of generations, 10,000 generations, if ever it would reach the wild type level. And finally, the DKA1, which has been in the collection in the clinic for more than 40 years, there is no indication that this bug will ever grow to become a real fast-growing Pseudomonas aeruginosa. So we has used this to actually study evolutionary trajectories, because by reverting from slow growth to fast growth, there is, and then sequencing the genomes and looking at RNA, messenger RNA synthesis, it is possible to actually trace back which evolutionary trajectories made these bacteria persisting. So we are trying to reverse evolution to understand forward evolution. That's the whole point of this. And the data is still not completely clear, so I'm not going to talk about it, but my next seminar in Argentina, hopefully on location, will probably deal a lot with this, because it's a fascinating way of studying evolution. We study it backwards, but we interpret it forwards. So this is a kind of picture of the metabolic specialization that occurs during a long time of Pseudomonas aeruginosa airways. And you can read the results out here. We see rewired fluxes of metabolites. So many different pathways, they are rewired. So things happen, so they don't produce the same with the same speed at least, or some are broken and some are open. We see auxotrophy, which is not surprising because there's a lot of amino acids in the lung substrate. So many bacteria lose the capability to synthesize amino acids. We see a change into low energy state and slow growth. And again, as I emphasized already in the beginning, slow growth is a very important diagnostic marker. Antibiotic treatment and resistance. I think all of you guys know much more about this than I do. I want to point out something which is very important. I showed this picture in one of my first slides. It is the complexity of the environment. It's not a test tube. But when the clinic determines antibiotic resistance of the bacteria, they take the bacteria out of the natural environment and put it into a flask with LB or with another substrate. And from there, they grow the cells, and then they measure sensitivity or resistance to different antibiotics. So the bacteria who they want to study the susceptibility to drugs from are taken out of their environment, and then they're analyzed in vitro in the clinical laboratory. And what they then do is that they translate this information back and say, this is what the situation is in the lung. And I have deliberately put crosses here across the arrow. This is not allowed. You cannot take the data here and trust that it's exactly the same susceptibility that you find in the lung. And we get a picture here which may suggest that we do have a serious problem. So here we have taken from all isolates from the early age infection of the CF patients and looked at resistance measured in the clinic, in the lab, to all the major antibiotics that are used in the clinic to fight Pseudomonas aeruginosa. And what we see is that essentially 80–90% of all the isolates are susceptible to all the drugs. Only a few seem to be resistant during the first, let's say, 10 years of colonization of the lungs. So this, of course, is contradictory, because we know that most of all these bacteria, they persist in the lungs. They are treated with antibiotics, but they are not antibiotic resistant cases. So persistence must be rooted combinations of environmental and organismal characteristics. We need to know the environment, and we need to know the phenotype of the organisms in order to explain the persistence which is not caused by antibiotic resistance. We have looked at high persistent variants. We find many of them. I'm not going in detail with the screen test we have done, but look here, 9% of the isolates we tested, they have a persistent phenotype like this, not like this. This is the normal wild type persistent phenotype. You treat, you remove the antibiotic, and then you see that you don't kill everything in the population. In a normal wild type Pseudomonas, you kill most, you kill all the way down here, but in a high persistent variant, you leave behind a very substantial increase in the population of those which are not killed by the antibiotics. And obviously this means that when the patients are treated with antibiotics, there is a great chance for these variants that they survive. And once the antibiotic is removed from the patient, they can resuscitate, and then they can go on. They can become really resistant at the end of the day. This is from the experiment rate, where Ruggiero did the reverse evolution, and what he picked up was faster growing bacterial strains. And when he measured the antibiotic susceptibility of these fast growing revertants, in essentially all cases, he saw that the tolerance to antibiotics was decreased as the bacteria grew faster. So in other words, if we translate that back to the state in the lungs of the patient, the slow-growing bacteria are more tolerant to antibiotics than the fast-growing bacteria. Because when you revert them to fast growth, they become more susceptible. So that, of course, might explain why there seems to be a selection for slow-growing bacteria. There may be many other explanations, but at least this makes sense. So altogether, this is our model picture at the moment based on all our paid studies and simple laboratory experiments. And what you see here is all the stuff that goes on with these bacteria from they enter the lungs and are very naive and very wild type. They meet the immune system, they meet antibiotics, they meet a changed nutrient composition, they have to fight for iron, they are meeting various types of stress factors from different, they go into persistent, they form biofilms, and so forth and so on. So basically, because of this long treatment period, and because we have these longitudinal isolates, and as we know how they're treated, which is indicated down here, colistin, ciprofloxacin, ceftazidime, topramycin, and so forth, we can begin to interpret this picture which happens here and over time from colonized to chronic, taking into account all the clinical information. And as I said before, this requires a lot of data integration, and we haven't really come very far with this yet. So now the last 5 minutes, if I still have 5 minutes, I just want to present our new program, which we got a very, very nice, handsome grant covering a period from this year until the end of 2025. And the whole point of this program is to develop improved infection models for studies of persistent infections. So realizing that test tubes are not really useful to analyze what goes on in the patient, we decided to reconstruct the patient in the laboratory, or rather, we reconstruct the lung. And in our case, with lung organs, we try to make miniature organs, miniature lungs in the laboratory, using stem cells or primary cells taken from the patient, and then developing them with different growth factors and stimulators, develop these stem cells into small micro lungs or miniature lungs, which we can grow in the lab, which are differentiated. So it's not just epithelial cell layers, but they're actually differentiated into basal cells and goblet cells and whatever you have in the lung. also immune cells. And we can, thanks to our collaborator at Stanford University, we learned how to change them from this apical in conformation into an apical out. Because if we have all the epithelial cells on the inside of this ball, cell ball, we would have to inject the bacteria and there would be limited space. What we can do now is that we can move these normally configured organoids into another substrate, and then they switch completely into this conformation where we have the epithelial cells on the outside. And then finally, we can also convert them into air-liquid interface. And what can we do then with this? Well, we can take from one patient stem cells and create the organelles. From the same patient, we can take the population of Pseudomonas aeruginosa so that we can reconstruct the current state of infection by simply adding the bacteria from the patient to the organoids of the patient. We can do single colonization. If there are many different types of Pseudomonas in the lungs, we can do competition experiments. We can add antibiotics to see how that impacts on the bacteria when they are in this miniature lung system. We can add immune cells to see how immune cells and bacteria communicate, and also how immune cells communicate with the epithelial cells. And we can use whole microbiomes. We can isolate the microbiota from the patient and superimpose that on the organoids and see which of these bacteria actually have a chance to prevail. And then we can look at gene expression in the bacteria, in the immune cells, in the host cells, and then we put it all together, and then some brilliant data analysts can make sense out of this. So we eventually can host pathogen markers, which are indicative of prolonged infection. This is how it looks like in the microscope. So here we have one of these organoid balls with tissue cells differentiated. We have green bacteria, we have red bacteria, and you can see they colonize the surface here. Here you see a little bit more detail in the confocal microscope and these bacteria have been stained with a fluorescent marker. This is not from our lab, so we borrowed that from one of our partners, but I'm going to show you our first homemade organoids from Signe Lolle, and she's the postdoc who is setting up this system. So this is our very weeks old, it's our very organoid picture showing bacteria, especially colonizing the tight joints between the cells, which is very, very interesting because we know that Pseudomonas aeruginosa actually very often goes to the tight joints also of plant tissue. And therefore, we are pretty excited about this first picture, but of course we need to see many, many more of those. This was just PA1 doing that, but it's just to show that we now think we are really on the track. So this is what we're going to do from this year and until the end of 2025. We're going to collect those, we're going to make libraries and biobanks, not only of bacteria, but also of cell cultures. We're going to do genomics, as we've always been doing, bioinformatics, but we're also going to do phenotyping both in vitro in the lab and in our organoids. And we're going to put it out into a facility we're building up for data analysis, so we can make translation of all this information, both to diagnostic tools, so it becomes easier for the doctors to diagnose the state of infection, and also, hopefully, to define new target treatment and interference. So with this, I will close. I want to mention some of the main characters here. First and foremost, of course, for many years, I've been collaborating with Professor Helle Krogh Johansen from the main hospital in Copenhagen. And she was the one who collected all these early stage infection isolates. And we have shared an office for 10 years now, and probably will continue to do so if we don't start fighting with each other. Rasmus Marmi is our old-time bioinformatician. He learned from scratch himself what bioinformatics is, and therefore he is a very clever guy with bioinformatics. Leah was a student of Rasmus in bioinformatics, and she has been working tightly with Jenny Bartel, who is an American in the group. And these two guys have been working on the statistics and the modeling of our data. Elio is the transcriptomic person who did all the courageous work on transcriptomes from the sputum samples. And Rogero is our metabolism expert in the group. Two Italians, he's from Milan, he's from Sicily. And finally, the new postdoc started 1st of May. And I showed her first organoid previously. Signe Lolle, she has a background in plant microbe interactions and also a little bit of cell biology. So with this, I finished my story. We are very excited about all this which is going on now, and I can certainly take some questions if you have some. Thank you very much.
Thank you very much, Soren, for another fantastic talk. Well, let's go with the question. Question open, okay. Diego, do you want to, okay, come on, Diego.
Hi, can you hear me?
Certainly hear you. I cannot see you, but I can hear you.
I can turn on.
No, it's fine.
It's okay. Okay. Well, first of all, congratulations for the presentation. It was really amazing and great. And for someone that have followed your work since started with biofilm and to see now to go into the patient, I mean, I think this is amazing and congratulations. And I think it has been also very honest from you to tell how difficult and how many trouble you have to tackle to really get you. And I think this is really nice for a student that sometimes, you know, yes, you get unmotivated when you see how many times or experiments fail in the mid you are trying to do. And so I think this is amazing, all the work, and think congratulations. And I think now to see that where all this going into recreation of these lungs, it's really highly promising. So I'm looking forward to see. So, okay, I'm just two questions. I don't know if I guess someone also have more questions, but I'm coming more from the stress resistance. And so one of question when you say that growth rate, it's important. One question is, so this is measured in vivo or this growth rate? Because I mean, once you take the bacteria, you put in the plate, you get nutrients, so they try to grow away again fast, but then I mean you get or they are stuck in really a slow growing mode.
Yeah, of course, pointing at something where I was running away from my own arguments, because since I argued that you cannot interpret in vitro data into the, well, but we actually many years ago, we did estimates of in vivo growth rates of Pseudomonas aeruginosa in a number of patients. And the way we did it then was simply to take samples, sputum samples, and look at the number per cell of ribosomes. So we used the classical relation between ribosome number and growth rate. And it works for E. coli and it works for Pseudomonas aeruginosa. And when I was a student, I was brought up with this kind of correlation. So I was reviving my studentship when we did these experiments. And they showed clearly when you compare to control calibration curves in the lab, that also in vivo, the bacteria grow very slowly. So,
okay, yeah.
Of course, many bacteria don't grow at all. You have a population of cells which are stationary, but those that do grow slowly if they are clinical strains.
So I don't remember the regulation in Pseudomonas, but I would imagine stress response behind and stringent response, or
yes, yeah, but it's the same. I mean,
it's the same
from that point of view. Pseudomonas is a pseudo E. coli.
Okay. Okay, and if I'm allowed the second one, I mean, it was for me quite surprising when you mentioned that community structure is not really or have a minor impact. When you that mean that other bacteria that form mixed community or come together, it doesn't seem relevant. There is a lot of literature or at least try to focus on this aspect of mixed community, staphylococcus, and so I'm
if we looked at the individual genes and their expression in individual cells, we would see differences. I mean, we would see impacts. So you know, the principal component analysis should be taken with a grain of salt. But by and large, what we're saying and what we're seeing is that there's not a huge difference in the way Pseudomonas aeruginosa expresses its genome, whether it's present alone in the lung or whether there are many other microbes in the lung. But if you dissect the genome expression, then you will see differences, of course. But for us, the important thing is to find a fingerprint, which the data I showed you were from old patients that had chronic infections. I'm pretty sure if we go to young patients and try to do the same analysis, we will see a displacement of, I mean, we won't see the same distribution in the PC plot. So what we want to do is to see if we can use this kind of analysis, the pattern, as an indicator of progression from naive colonization to chronic state of infection. Because if it only takes a couple of RT-PCR analyses, it not be impossible to do that in the clinic. And the major problem in the clinic is they do not know what to look for in terms of indicators of switch to a adapted state. So we're trying to help them with that. And this analysis has to be repeated. We started with a chronic infection because the bacterial biomass is so much bigger. But eventually we have to go down and we should be able to measure on 100 bacterial cells the transcription profile. But we are not there yet. It's a little tricky. So
no, I understand, but it's certainly very interesting. Well, I thank you very much for your answers. Very interesting.
Okay.
Thank you.
The next, Mariana Puri. Next question, Mariana.
Yeah. Hold on, I'm trying to start my video. Let's see. Hi, how are you?
Hi. Hi.
Thank you very much for the wonderful talk. I really enjoyed it. I have a question, and actually it's more a comment, but I think what you showed today, it makes total sense now, and I really like the phenomic approach that you are using, you know, because I think we cannot rely in just genomics to see what's going on, especially in a patient, no? And I really like your results about the growth rate in these strains and how the growth rate is decreased. And of course, it makes sense that with a slower growth rate, the response to antibiotics, it's not gonna be optimal because most of the antibiotics that are used in the clinic, they rely on a replicative state of the bacteria. So I'm not familiarized with the kind of antibiotics that are usually used for treatment of these infections. But do you think that maybe a way to go to treat these patients and these kind of infections is try to find actually antibiotics that can act in a non-replicative state of this bacteria after the results that you showed us?
Yes, I think some of them actually do. For example, colistin is actually killing stationary cells quite efficiently. And colistin is a very popular antibiotic in the CF clinic. So together with Cipro, which of course is a broad-spectrum antibiotic and which only kills growing cells, that is a very frequent combination. So very often the treatment is combination therapy. Taking 2 or maybe even 3 antibiotics. at the same time. So you're absolutely right. It's essential to either use drugs that also kill non-growing cells, or to treat for very long periods of time, because those cells which are stationary at one point will eventually start growing at another point, and then there should be the antibiotic present to kill them. But then you also increase the risk of resistance development. And so all the time, it's a race. It's really a trade-off. You don't want to increase too much resistance levels, and yet you want to eradicate as much as possible the bacteria. And I think that's the dilemma that the medical doctors have all the time. And I'm actually not convinced it will ever be solved. I think what we're not trying to come up with a miracle treatment or miracle suggestions, but we do think that they can be smarter in the way they treat if they know what's going on. And therefore we hope we can prolong the lives of the patient. So instead of dying at 40 or 45, maybe we can get them to 70 someday. If I think it's a matter of knowing what the hell you're doing. And very often it's like picking drugs from the shelves, and I have a very hard time understanding the rationale when I look at the treatment strategies. Mm-hmm. There may not be one.
Yeah. Thank you.
Arlinette?
Okay. Well, thank you very much for a very nice talk. I just want to ask you regarding the slow growth and the persistence. It's also related with growing in biofilm.
Yes. So it's true that if you have a biofilm, you have many more persisters. That Kim Lewis showed that a long time ago. And basically, what we hope to do, and that's actually a collaboration we did with Kim Lewis, we hope to see that specific genes were always mutated in the high persister variants that we pick up. So that was the whole purpose of the project. But when we analyzed the high persisters, which survived much, much better during treatment, there we found mutations all over the place. There was no pattern whatsoever. So our conclusion is that there are many different ways that bacteria go into a dormant persister state. It's not just one or two or three. It may actually be combinations of gene mutations epistatic effects, which put them into this state where they can no longer be killed because they are high tolerant to the antibiotics. So from that point of view, it was disappointing to the persister community, because they were all strongly believing that there was, smoking gun mutations that would show up in analysis like ours, and we don't see. But nevertheless, it's important that you do have a high fraction, 20% of all isolates, which we picked randomly from a patient, have this high persister phenotype. So the phenotype is clinically important. And again, as we talked about before with the previous person who asked questions, if we could then develop some antibiotics that would kill persisters, that would be a really advantage, an advance if we could do that. So in the center where I'm working, we have a group, very large group, that try to discover antibiotics. And we have been talking with them about we can deliver persister cells, and then they should try to take their new antibiotics and see if they can kill persister cells, because the conventional antibiotics don't do that.
Thank you.
Sure.
Okay. Julia?
Hello, good afternoon. Well, first of all, I wanted to thank you for an excellent presentation. It was very clear, and especially for people like me that I'm not in the subject, so it was very, very clear for me. And I found it very interesting that you found slow growth to be the most dominant characteristic. And what I thought is, could this be a strategy to evade the immune response? So have you or anyone analyzed the immune response to the different strains, and especially to slow growers?
This is a great question. I mean, I cannot answer it because we even didn't think about that. But I think we can try that out now in our new model system, because we will look at the impact of immune cells and then take different types of bacteria and see how they interact. So you could probably be right about that. And also, as I tried to say before, the increased tolerance to antibiotics is just one advantage of the slow growing cells, but I'm convinced there are other advantages. But maybe the suggestion you come up with is the most important one. I don't know, but of course we will see that when we start working with our system. But thanks for the suggestion. That's a good one.
So maybe they can keep under the radar in some kind of way.
Yes, no, it's a perfectly good suggestion, absolutely. And of course, the immune system is normal in these patients, so they are not immunohandicapped. So, yes, good point. Yes.
Okay, and next, Alejandro Moyano.
Well, thank you very much for such a great talk. My question is there any correlation between those clones that are more epidemic and the growth rate, or what can your phenomic approach tell you about those clones that are more epidemic and their phenotypes?
Yeah, we are still searching. It's a very interesting question, of course. I think it has been shown, certainly by other groups, that when you take from the environment a lot of different clone types of Pseudomonas aeruginosa, and then see which of those you frequently find in CF patients, they are not equally frequent. So there seems to be some kind of pickup in the different clone types, which make them either more potent in colonization and persistence or not. And we from the little cohort that we have with 35 patients, and we have about 60 different clone types, we have already seen that some of them, when they infect, and they infect more than one person, then they tend to persist. So yes, we see the differences. One of the things we're going to check now, and that's a new postdoc we just hired from Chile. She's going to characterize the accessory genome, because nobody has cared about the accessory genome. So there may be something in the part of the genome which varies a lot from strain to strain. And we will see, hopefully, at the end of this, whether they participate, these genes, which are so variable in the persistence and colonization and success on the bacterial side. So there is something going on. I just cannot answer specifically what it actually is, because reading the genomes is not, it doesn't tell us anything.
Thank you.
Okay, the next question is from Dr. Soledad Celes, but she has not a mic available, so I'm going to read her question. A naive question from a protein biophysicist far away from microbiology. A few years ago, the operon Fab was identified in Pseudomonas, including Pseudomonas aeruginosa. It encodes for a functional amyloid protein, FABC, and accessory proteins. FABC is involved in biofilm formation. Have you tried bacterial resistance using a combination of antibiotic and some anti-amyloid drug?
No, but it reminds me of something which is also very interesting, and I don't know whether there is a connection here. So we have been involved a little bit in a phage, Pseudomonas phage investigation, where one of the most common bacteriophages in the genome of Pseudomonas aeruginosa seems to play a very important role in terms of successful colonization. And that particular phage is a single-stranded DNA phage, which when it comes out in the environment and interacts with polymers, it creates crystalline water. And I have no idea, I'm not a biophysicist, I have no idea if amyloids somehow have the same capacity to interact with polymers in the same way. So the question back to the person who has asked this question would be if this particular protein she's talking about, or he is talking about, could be phage produced, because I think we have missed entirely all the bacteriophages that we know interact with Pseudomonas aeruginosa. And we recently started a Greek postdoc on a phage project. And she's going to figure out if phages, both lytic and lysogenic phages, can impact on the resistance to both antibiotics and immune system, and also to the success of colonization in our organoid system. So that kind of physical conformation of proteins is really, really intriguing. And the only case I know about is this phage interaction of the monomers that gives crystalline water. So everything is really put into a matrix automatically. But it's a fascinating stuff. Yeah.
The next, Alejandro Viale.
Okay. Okay, thank you very much. Very nice talk. I think you have already answered my question because I was curious about the persisters. I mean, I would have thought that you would have found a couple of genes because it seems a very stable phenotype, but you already said that there is no particular gene mutated on them. Have you tried whether there is some permanent auxotrophy in all of them? This is one question. And the second question is, what about the other bacteria? I mean, not Pseudomonas aeruginosa, but the other bacteria. I mean, you find also a lot of persisters there? Or have you tried whether there are a lot of persisters in other species other than Pseudomonas?
Yeah.
No, third question, sorry. How do you kill persisters then? I mean, how do you eliminate them?
Let me just clarify with respect to the genetics. All I'm saying is that it's not just one or two specific mutations that give the high persister phenotype, but I'm sure that mutations are responsible. It's not just a random phenomenon. It's not a stochastic phenotype, because it is stable. So when you take those clones with high persistence, they maintain their state of high persistence. So we don't know what the mutations are or what combination mutations, but of course there is a genetic cause of this. I'm not sure I understood your first question. What was the first question?
This was the first question, where you have picked up the genes responsible for this. And the second question, whether
that's about the other bacteria.
If all the persistence, all the persistence, whatever the mutations are, or they show the same auxotrophy for amino acids, for instance.
Yeah. We haven't done that correlation at all because we never thought about that would be interesting, but it might be. Concerning the other bacteria, at the moment, because we worked with such a complex system, we forgot about everything, which was not Pseudomonas. But now we want to bring them in. So we really want to study microbiota from the lungs in the kind of models that we are analyzing. And I think it's doable because several people have done that also with the gut. So when you make spheroids from the intestine and look at the intestinal microbiota, you can actually handle it in the lab. So I cannot answer your question, but we're doing something about it.
Okay. And how do you kill the persisters now? Yeah. I mean,
how to
what
kill
are
the
like,
resistance. Yeah,
yes.
it's a good
So
one.
you mentioned that you're working on that also.
We will see if we can by chance get something. At least, you know, if you direct those people who look for new antibiotics into a certain direction and say, forget about biofilm, forget about whatever, and you have a collection of persistent cells which we have sorted out somehow, and if you can kill them, we have something which is wonderful. So that is the challenge for them, and we'll see what comes out.
Okay, thank you very much.
Sure.
Adela?
Hi, thank you very much,
Hello.
Soren, for hello. Well, my questions were also about the genetic basis of the persistence, but I would like to ask you if you have any data of patients that has been treated with these new therapies for CF that revert the phenotype, or what is your opinion about how this therapy will impact?
Actually, Jane Patel, who was the American in the group, I showed the picture of her. She got her own grant from the American Cystic Fibrosis Association, and that was to study the microbiology during treatment with these promoters or not promoters, the modifiers via molecules. I think in Copenhagen, at least, the conclusion is that these modifiers don't do very much to the microbiology. So they rescue something like 10%, at least those that have been used in the clinic. They save 10% of lung function, and the microbiology is basically the same. So from that point of view, it has been a little bit disappointing. On the other hand, I think that's the only way to save the patients. That's to get the CFTR to work normally again. And of course, we are working mainly with the first or even, well, maybe second generation compounds. But when we get to the 5th, 6th, and 7th, and 10th generation compound, they may actually be able to synthesize or find something which restores 50% of the lung function or of the CFTR function. So I think that's the point. It's a good strategy, but they are pretty far from the goal, and we cannot easily read it in the microbiology.
Okay, Lucila, I don't know if you want to read your comment. It is a comment from Lucila, or do you want that I read the comment, Lucila?
Yeah, I can read it, Andrea.
Okay.
Thank you very much. Thank you, Dr. Molin. Yeah, I'm from Tucumán. I'm a microbiologist, but my 13-year-old girl has CF, so I'm also into the Pseudomonas and everything. Yeah, but I'm working on the gut microbiome. But just a comment that I totally agree with you with the persisters and the. For clinicians and medical doctors, it's so important to detect the persistent, and they know that they are persistent and they cannot treat with a simple antibiotic as they are prescribing, because you know what's exactly what you told. And also just a little comment about the modulators. I know the lung function is improved 10%, but they report that there is less Pseudomonas exacerbation. So have you checked any Pseudomonas from these patients that are treated by, with, for example, Trikafta, which is the new one, very famous one?
I think what you have to keep in mind is that the treatment in Copenhagen, in Denmark, of CF patients really is very different from many, maybe most other places. And therefore, treatment is so aggressive with antibiotics and efficient that add something from later is not going to make a difference. Whereas if you go to other clinics around the world where treatment is so systematic and aggressive, then you may see these much more easily. So I think that could explain why we haven't seen any exciting differences. But it's simply because at the moment and since a long time ago,
yes, I know. I have read, and also
so efficient.
Yeah. And they have the highest rate of survival there. So because of that,
exactly.
yeah. Okay.
And that's by and large, it's because they are good at removing Pseudomonas.
Yeah,
also.
of course. Thank you very much. Excellent talk.
Sure. Thanks.
Thank you.
Wow. I last question, because I know it is very late in Denmark now.
I have to go to bed. I'm an old man.
No, I'm about reverse evolution says that you talk that you recover clones that are able to grow faster and to also to that are more resistant after this reverse evolution.
More sensitive. They're more sensitive.
More sensitive. Okay.
Yeah, when they grow faster.
Okay. Anyway. Okay.
Yeah.
Did you sequence these clones, or are you analyze the mutations that are based?
Yes, very interesting. Andrea, I'll send you we have a manuscript now, which is in review at Nature Communications. So I'll send you the manuscript. It's very interesting. And you might, we also did transcriptomics. So we also know which regulatory mutations may be involved. So I'll be happy to send the manuscript to you.
Perfect. Okay.
Because I don't remember right now all the different types.
Yes, obviously.
Some surprising mutations in gDNA, and other DNA metabolism mutations, and then there were regulatory mutations also. But it's a whole range, and it was a little bit of a, as usual, it's complex. So
perfect.
But
okay,
I'll do a paper, sure.
Thank you very much. Before finishing, I like to ask all to switch on camera in order Matthias can take some pictures to remember this nice occasion.
Ok, vamos. Una. No sé qué fin de pantalla. Y la última. Perfecto, gracias.
Thanks, Matthew. Okay, I like to thank you again very much, Soren, for sharing all these investigation and project with us. It was a great talk, as usual.
Thank you very much. My pleasure, all the way.
Very much.
So, all you
Thank
guys,
you very much.
Hi, Soren. Okay.
Take care of yourselves.
It was great. Thank you very much.
Hi, Soren.
Thanks, Reading, for.
Thank you.
Good night.
See you soon, Antonella.
Bye. Thank you for the great talk.
Bye-bye.
Bye.
Bye.
Bye.